Okay, so now let's move on to transcription. And so here's where we're going to use again DNA as a template to make some RNA, and it would be correct to say that we are going to essentially transcribe a gene, a particular section of DNA that's responsible for making a particular RNA that then will either be used to make a protein, or that RNA will serve some other purpose inside the cell. Alright, so we're going to refer to also that gene is known as the transcription unit. Now we'll see this process is quite a bit simpler than DNA replication, in that there are not nearly as many enzymes involved as there are in DNA replication. So the primary enzyme involved here is what's called RNA polymerase, and it's going to do most of the work here. And so it's going to, as we'll see, attach to a part of the DNA, a part of the chromosome that's just, you might say, upstream of the transcription unit at the very beginning of it. It attaches there, it unzips or unwinds the DNA. It starts building the RNA by adding complementary bases. And you see here that attachment phase is what's called initiation. Then elongation is when it moves along, adding the appropriate bases. And termination is when it reaches the end of the transcription unit and the RNA polymerase drops off, and you have your RNA transcript made. Now let's look at those steps a bit more detail here. Now initiation, again, it's going to attach to the promoter that's at the beginning of the transcription unit. And the promoter has this particular section of bases in it, T's and A's and T's and A's, and it's sometimes referred to as the Tata box. So that's sort of right at the beginning of the promoter, and that tells us where the transcription should start. Now, RNA polymerase does get a little bit of help in that these proteins known as transcription factors attach to that promoter, and so each gene will have specific transcription factors that basically signal it's time to transcribe that gene. So those transcription factors tell RNA polymerase where to go, where to attach, and where to start doing their job, where it starts doing its job. So then elongation, the RNA polymerase moves along, it's reading the template strand, adding the appropriate complementary base, again adding U's instead of T's, until it finally reaches the end of the transcript and it finishes making the RNA. Now this is from a little later in the chapter, but this shows us that you can have several RNA polymerases moving along the gene, transcribing several copies of the RNA at the same time. And this also shows the next step, the translation step, and how each of these little brown blobs represents a ribosome, and how you can have multiple ribosomes then also working over that RNA transcript to make multiple copies of the protein. Alright, so what will happen is the RNA polymerase will reach the end of the transcript, and there's a particular sequence there essentially that sort of signals, okay, this is essentially the end of the transcript. And so the part that's shown in red here with this little other boxes here, this red part is essentially that RNA transcript that is being made. Now we'll see during processing what's called the cap and the tail are added. They have particular functions when it comes to translation of essentially the 5' cap tells the ribosome, well, this is the beginning of the transcript where we start, and the poly A tail serves as a signal that this is the end of the transcript. These caps and tails also serve the function of sort of being a protective end on the transcript to sort of protect those inner base sequences from being degraded. Alright, now let's go to RNA modification. So, like I said, the red part essentially represents our initial transcript. Again, we add the 5' cap and tail to protect the ends and serve as signals for the beginning and the end of the transcript. Now, within the part that was transcribed, we have sort of some stuff we want to keep and some stuff we don't want to keep. Okay, so we have in that transcript what are called exons and introns. The exons are the parts we want to keep. The introns are the part we want to cut out. And the number of introns and exons on a transcript will vary for any given gene in a eukaryote. You can see several of each. But in each case, we're going to want to remove those introns, reduce the number of bases so that our final transcript, as you can see, is quite a bit shorter than our initial one. We're going to cut out quite a bit of it. We're going to retain the cap and tail, though. Alright, so how are these introns removed? Well, that is the job of the spliceosome. The spliceosome is sort of an aggregation of proteins and RNAs that get together and help cut out the introns. So you have these small nuclear RNAs that get together with the particular protein, and these things together are known as SNRPs. And so each SNRP here also gathers with other proteins, again, to make this thing called the spliceosome. And its job, particularly using those RNAs, is to identify the base sequences that represent the introns. And then once those have been identified, the spliceosome essentially cuts out the intron, and so this extraneous nucleic acid would then just be recycled and used in other nucleic acids. And so after this, you're just left with the exons. Okay, now remember that prokaryotes essentially don't have introns and exons. You might say their whole genome just consists largely of exons. They don't have to splice anything out. So you might ask yourself, well, why do eukaryotes have these introns? Well, that's a good question. So we'll see that having these introns allows, and this is described on page 336 of your book, what's called alternative RNA splicing. And that is, we'll see that the initial transcript, so a particular gene can have different introns and exons. And so under some conditions, some portions of this transcript, as is shown here, might be exons, but in other situations we might say have an exon here. And so we have what's called alternative splicing, where we put together different combinations of exons. And we'll talk about the reading frame and how the reading frame can be different, leading to different combinations of exons. Now, these exons often represent, as is shown in figure 1713 of your book, which I don't have on this slideshow here, the different domains. You remember we talked about the fourth level of protein synthesis or structure, where you take different subunits, put them together. So these exons can represent different subunits. Also, the introns allow for, when crossing over occurs, that it decreases the likelihood that mistakes will be made, because as you can see, this particular gene consists largely of introns, and so if crossing over occurs here and somehow one or two bases ends up missing, it's not a big deal. Not that that will necessarily happen when crossing over happens, but it can happen. So if the crossing over occurs in an exon, it just increases the likelihood of a mistake being made. And of course, in bacteria, crossing over doesn't occur, so that's not really an issue with them. Okay, so let's see. I'm going to save translation and mutations for one more video. Okay.